Well, thank you very much for those kind words and really for inviting me to this oncology group meeting. I'm sure you know that I don't really like to travel much after this COVID, but I honestly could not say no to both Sujit and also Swapnil when they said, I could come here to. Oh, of course. Yeah, he's always wanting to make himself prominent over here. But anyway, thank you all very much. It's an honor, really, to come here and deliver the first oration in the name of Dr. Agarwal. I must confess that I really didn't know Dr. Agarwal at all well. Of course, I'd heard of him. And this is very interesting to know that he was the one who started the slide seminars at MapCon, which I can tell you are really, really very popular. So if you want to fill the hall at any of the MapCon conferences, then, have something where the slide seminar is placed such that people can't leave the room or are there waiting for the slide seminar, and so, the speaker gets a good audience. When they asked me to talk on, give an oration at my age, what can you say except to give a little bit of philosophy, something that you may have thought about over your long years of working in the field. And I have done this before. But I thought that I should repeat it because I think it bears repeating. I hope this doesn't sound very pompous, but I have had a really long time to think about whether everything that we knew or thought we knew about cancer all those so many years ago is still very valid today. And perhaps if we really introspect, you will find that there are many things that we do, not just pathologists, as well as clinicians as well, may not actually represent what you thought, what cancer really is. And I think this is something that bears thinking. So let me just tell you what my understanding of cancer was 40 years ago, and each one of you sitting over here, just think whether this is still your understanding of cancer. Some of it may be very valid, some of it may not be. Now, here you are. Cancer, of course, is a malignant neoplasm, and I think we do believe that cancer is neoplasia, okay? And what is this definition of a neoplasm? And I'm sure I see lots of people who are younger than I am over here. I must be one of the, the older lot that is over here, but even younger than I am, who still can recite this definition, yes? And what is it? That a neoplasm is an abnormal mass of tissue, the growth of which exceeds and is uncoordinated with that of normal tissues, and which persists. It persists in the same excessive manner after the cessation of stimuli which evoked the change. You see me smiling, because you see who defined it that way? R.A. Willis. And somebody of my vintage knows that R.A. Willis was already dead when I was, a postgraduate. So you know exactly how old this definition is, okay? And then, of course, following on from all of the research, etc., that was done in the early years of the previous century, we came to this understanding, certainly it was there in the textbooks that I read for a very long time, is that cancer is a multi-gene growth disorder. It's not a single gene disorder. You find lots of genes that are abnormal in this in the cancer. And this growth disorder is caused by somatic mutations which arise from the clonal expansion of a single neoplastic cell. So there's one neoplastic cell, it gets mutated or its genes become abnormal, it goes rogue, and you have a clonal expansion of these cells. Just think about it. Think of all the treatments and the management that we do based on this understanding of cancer, okay? And when you get a growth, these cancers exhibit defining hallmarks, and this is, the Weinberg and Hanahan, or Hanahan and Weinberg hallmarks. You just have to open any textbook, and we are all, or many of us are pathologists over here, and we teach that to our students. All the textbooks, including Robbins, which is supposed to be the Bible that we expect our students, postgraduates or otherwise, to know, mentions that, and you'll see some very lovely pictures, diagrams based on this, okay? And all of this is based on this one fact, that there are consistent differences between normal cells and cancer cells. And a cancer cell is definable by and of itself, okay? And I think those of us who are in this business of oncopathology must believe in it, otherwise how do we make diagnoses of cancer? But just let's ask for a minute, okay? This definition of a neoplasm, which I've put up here, the Willis neoplasm is on top, and then, Robbins has added to it, to expand on that definition. It says, fundamental to the origin of all neoplasms are heritable changes that allow excessive and unregulated proliferation that is independent, again, of physiologic regulatory stimuli. I haven't made it up, I've copied it from Robbins' textbook. Now, if that is the case, is a uterine leiomyoma a neoplasm? And I'm sure all of you are going to be aghast. Of course it is a neoplasm. Really? If this definition is true, do you think that leiomyomas would shrink once menopause came in and the estrogen drive had disappeared? And what is that estrogen drive if not physiological regulatory stimulus? Okay? Yes, it is abnormal. Yes, it persists. Yes, it is uncoordinated with that of normal tissues, but it is certainly not independent of physiologic regulatory stimuli. But you and I know, okay, despite all these definitions, that a leiomyoma is a neoplasm. Yeah, I think we agree. I don't need to take a poll of, people in this audience. So it is a neoplasm. But it's an exception to the rule, but if you just think, I just said leiomyoma, but there are many exceptions to this rule of physiologic regulatory stimuli. You know that we cut off the estrogen drive, and you know what happens to breast cancer, all right. But if we are going to give a pass to a leiomyoma and say, yes, it is a neoplasm, but you cannot apply everything to everything, then tell me, what about an extra thumb? Is that a neoplasm? It's abnormal, it persists. And then you say, but it does stop. Yeah, but then so does a leiomyoma, but you said, hey, give a pass to a leiomyoma. So are you going to give a pass to the extra thumb? You just look at the definition. And then the question arises, if you think that a leiomyoma is a neoplasm, and yet everybody in this audience, if I ask them, do you think an extra thumb is a neoplasm, what would you say? The answer would be, no, it isn't. Yeah, now I don't want to confuse you, but what is the difference? And the answer is simply this. It is about tissue organization. That is the missing part of the puzzle. When you look at a thumb, it may be extra. You look at an extra lobe, whether it's in the lung or it is in the liver, it is organized like you expect that particular tissue or organ to be organized. But when you look at a leiomyoma, tissue organization is definitely disrupted, okay? So there must be in that definition somewhere which has not been explicitly mentioned that a neoplasm must have disorganization in the tissue. All right, so let's start putting down some of these thoughts. It is uncoordinated proliferation. I mean, I don't think that that's in doubt when you're talking about a neoplasm. Yes, there is disordered tissue organization, okay? And there, we add this often, that there is a clonal, it's often a clonal process, and you know what is a clonal process, that it affects one type of cell, and it can be traced to one progenitor cell. So just out of interest, I once gave this, talk to students when I was teaching undergraduates and also when I talked to postgraduates. And then I asked, if this is a clonal proliferation, can an amoeba get a neoplasm? And I'm just asking, I'm delivering a lecture, so I don't want to engage you people with a yes or a no. But very recently, when I talked about this, not even 2 weeks ago, a very senior pathologist told me, yes, an amoeba can get a neoplasm. And so in your own minds, poll yourselves and ask, if it's a clonal proliferation and one cell gives rise to a whole clone of cells, can an amoeba get a neoplasm? And the answer is no. It is unequivocally no. Why? Because if you look at that definition above, there is a story about tissue organization, and an amoeba is not part of a tissue, okay? An amoeba is a single-celled organism. So, to everything else, you will have to add this part of the definition, and that is, neoplasia is a process that can only occur in metazoa, where metazoa produce tissues. There are many metazoa that don't have tissues, okay? You can think of sponges and several other of these kinds of animals that don't produce tissue organization. So you cannot get neoplasm in an amoeba, okay? You can only get it in metazoa. So if you have to just repeat that, and this is the definition that I like, and I always tell young people who are postgraduates, please not to mention this definition if they are asked to write a paper on it or to answer it in their vivas, because people would probably wonder where did they get this weird definition from. But I believe that this is as close as possible that you can come to a definition of a neoplasm, right? It's not the Willis one. I don't think it is the Robbins one. This is, well, I've concocted it, but out of what I feel is essential to understand what a neoplasm is. And when we go down the line, you'll see why it makes a difference, particularly this issue of tissue organization, which is essentially about differentiation. So it is an abnormal, often clonal, cellular proliferation that exhibits uncoordinated growth and differentiation in a multicellular organism. This is one of the insights that I have, now that I've been in this field for so long. So let's then ask, if this is the definition of neoplasia, what's a cancer? And of course it is a malignant neoplasm, okay? But how do we know that something is a malignant neoplasm? And the only way we can know it is from its behavior. We have encountered that behavior before in a particular setting where something looks like it does. So there is no way that you, I, or any of you sitting in this room will make that diagnosis unless we or people before us, that we all stand on the shoulders of giants, have said to us, told us, or we have now come to understand that when you see something that looks like this, it is going to do two things. It will invade and it will metastasize. That's the only way that we know that something is a neoplasm. Although day in and day out, we make diagnoses of benign and malignant, we are doing it from prior experience, which we correlate with its morphology, its immunohistochemistry, its molecular, today increasingly its molecular behavior, but there is no intrinsic way that you can say that something, because it looks like this, is a malignant tumor, is a cancer. And I put it up over there. You can see one of the ugliest looking, malignant looking tissues is a trophoblast. I'm sure you will all agree with me, yes? But it is not a tumor, number one, and it isn't, it's just because it looks like that, it's not malignant. And anybody who has seen a tubular carcinoma of the breast, okay, is one of the most quiescent, benign appearing tumors. It is just, we know that this is not a sclerosing adenosis, it is a tubular carcinoma, although everything about its organization, the fact that it's not, it doesn't look bad, there's very little to choose from it except organization, and that too tissue organization that tells us that this thing is benign and tubular carcinoma is malignant because it behaves like that, okay? So it's not about looks, it is about behavior. So let's get to away from this definition and let's take this. Okay, cancer is a multi-gene growth disorder caused, okay? That's very important in all those definitions or our understanding, caused by somatic mutations which arise from the clonal expansion of a single neoplastic cell, and this kind of growth exhibits defining hallmarks. Okay, let's examine that. And this is based, obviously, on this theory which is called the somatic mutation theory. And it is, for all of us, for all cancer research and treatment today, it is the paradigm for oncogenesis and tumor progression. I'm sure we all teach it to our students, and there are probably many teachers of pathology or past teachers of pathology that are in this audience, okay? And once again, it talks about these heritable changes that occur in the genome of the cell, and that change is passed on to the progeny of the cell, and then you have a clone of descendants, and that constitutes the cancer, and it displays all the hallmarks, okay? And over time, mutations accumulate, and these mutations lead to tumor progression. I don't think there's anything or anybody will dispute that this is our understanding of what is happening in a tumor. So once again, cancer is this multi-gene disorder that begins as a clonal alteration in one cell. And I don't need to tell you that we do this every day. And this is just a diagram, and it's taken straight from Robbins' textbook. I've acknowledged that down there because that is chapter number 6, neoplasia in Robbins. And it tells you this, that you start with normal cell, it gets initiated, okay? And that initiated cell, and I've highlighted it over here, has stem cell-like properties, which means that it's able to divide, and it's no longer under the control of the physiologic stimuli, as Robbins was wont to say, okay? And then, of course, they get some driver mutations, and I'm sure, I'm not going to tell you what these all are, and finally you get many more mutations, and you get a clonally heterogeneous tumor. It starts as one cell with which is abnormal, then the progeny accumulate other cells because we are told that cancers are inherently genetically unstable, and you get a clonal progression. I don't want to belabor this point anymore. And this has been shown time and again, but really the most seminal paper on this clonal heterogeneity within a tumor was Gerlinga, which was published in the New England Journal of Medicine quite some time ago, where, the people looked at this particular group took samples from 4 renal cell carcinomas, but took it from different areas of each of these tumors and compared them to each other. That means not between tumors, but between the different areas within the tumor and also from their metastases. And they found that it was almost like looking at Darwinian evolution. Okay, you have You have some, like the VHL gene, which is mutated because these were all renal cell carcinomas. And you found that as you diverged from that, what you call a stem line, you got variegated mutations. And you can see in this one tumor that they're showing you over here that there is a branch. Okay? And then each of them goes branching in different directions. So there is clonal heterogeneity, and this somatic mutation theory is coming, is being fulfilled. You're getting clonal expansion. We use this today in our in investigation of tumors, particularly when you're looking for resistance mutations in targeted therapy, by doing liquid biopsies. Why? Because tumors shed cells, they shed DNA, which you can use, like cell-free DNA. And because there are so many metastases and there are so many different parts of the tumor, you can't go biopsying them all. You get a comprehensive genomic understanding of what's happening in that tumor. And you can even look for why this tumor is no longer responding to therapy because of, you find resistant mutations in the tumor. You may not know which of those metastatic foci or which part of the tumor is showing you the resistant mutation because you're just doing something from the blood, but that is based on this clonal heterogeneity and the shedding of these cells and DNA in the blood. And we're using it now. There's this very famous paper that has now just been read at the last ASCO. They've been doing this for a long time, and it shows, if you are any colorectal cancer surgeons over here or those interested in colon cancer, that if you have to make a decision in a stage 2 colon cancer whether to give or not to give adjuvant chemotherapy, you can probably do it on the basis of finding out whether there is still cell-free DNA from the tumor circulating after 6 weeks. And if there still is, it means that this patient is at a very high risk of developing a metastasis. That has been published now, and that's the particular group that probably will benefit from giving chemotherapy. So far, so good. And here we have the hallmarks, and you see here there's Hanahan and Weinberg, who are really the high priests of the somatic mutation theory. These are the 6 hallmarks that they have published before. You can see all of them over here. I'm not going to repeat them because everybody who's a pathologist will know about these hallmarks. That was done in 2000, I think it was 2001, and they published again in 2011 4 additional hallmarks. Okay, one was, by that time immunology and of the tumor was becoming very common, and so they said, evading immunogenetics, in other words, and down regulating their cancer genetics. They talked about the microenvironment over here, as you see, and they talked about genome instability in the tumor. And this is what has been happening since 2011, when they published this, right up to 2022. There are innumerable therapeutic targets that have now been based on this somatic mutation theory and on the hallmarks. You see that? I mean, you just have to look at it. You just look at the hallmarks, and you have got a therapy that is directed against the hallmark, not against just killing the cancer cell. It's not just radiation therapy or just chemotherapy or extirpation, like we used to say in the old days, slash, burn, and poison. No, it is now targeted to the understanding of the hallmarks of this tumor. Well, so somatic mutation theory works. That's what we have to understand. Or does it? Okay, there are nagging questions with this somatic mutation theory. And let me just bring up a few. I'm not going to talk all about that, But one of them is the origin of therapeutic resistance, especially to drugs targeted at driver mutations, okay? EGFR, you target a drug to EGFR, okay? And then it becomes resistant. And where do you find that resistant mutation? Is it in some other gene that prevents the... No, it's in the same EGFR gene. And it's not on the gene that already has the driver mutation. Otherwise, where was the question of it not responding? So what's that? What is actually happening? All right, the other thing is organ specificity in inherited cancer syndromes. We know about, say, let's take retinoblastoma. You know that there are families in which you have retinoblastoma inherited. Why is it only every cell in the body has got the mutated or the abnormal gene? So why is it only that you get a retinoblastoma? Or very seldom you might get an osteogenic sarcoma or one other sort of sarcoma here or there? Why? Why doesn't every cell in your body, if it is somatic mutation theory and there is a mutant and that makes that cell, more susceptible to other mutations and there is progression, then why only retinoblastoma and occasionally an osteosarcoma? Just ask yourself this question, what explains that? All right, the other thing is that if this is going on, and cancer cells, once a cancer cell, always a cancer cell, then how do you explain normalization of cancer cells? And I think we find this every day, we can talk about it. And cancers can be induced by transplanting normal cells. It's not a cancer. And you put it in an abnormal location in the same animal. Of course, you can't put it in me and try it out. And they become cancers. And those cancers can be normalized by reimplanting them into the natural habitat. I'm not joking about this. This has been known for years, but it has never been explained by the somatic mutation people, okay? And then cancers that respond therapeutically, of course we know that about ER and PR and all, to physiological stimuli. There are also normal individuals who harbor cells, and the most common one that is talked about is the 14-18 translocation for a follicular carcinoma, a follicular lymphoma. At least 4% or thereabouts, depending on how hard you look, of a population. In the West, it's even higher, it's about 10%, but maybe in ours, because we don't look for it, are running around with the 14-18 translocation in their lymphocytes. But they don't have a follicular lymphoma, and many of them never go on to get a follicular lymphoma. So what's this story about? The fingerprint of a follicular lymphoma in your lymphocytes, and you don't get a follicular lymphoma. There's something else that is going on. Okay, cancer dormancy. The medical oncologist has dealt with it, the cancer has disappeared, and what do? Where did it go? Why did it come back again after 20 years? And then also you have to explain this, why cancers predominantly occur in adulthood, although most mutations occur when you expect it to occur, during fetal life and in early life. And yet, very few cancers occur at that age. It all occurs in adulthood. So we have to have an explanation. Now, therapeutic resistance in cancer is a little bit like what you get in resistance in, when you're treating bacterial infections, for instance, okay? You cut down the number of cells that are sensitive to whatever drug you're giving, and then there are these resistant clones which are lying over there, which then start to proliferate because the drug does not act against them. So everything goes down, and then suddenly something comes up. So we have to ask, how do these subpopulations arise, and where do they come from? And maybe we have to think that cancer is not really a uniclonal disease, or what we call monoclonal disease, or a clonal disease as the somatic mutation thing. Why should we believe that all cancers just arise from one cell? Is it not possible that there is multiclonality in the tumor? And the answer is, oh gosh, there are any number of cancers when you go to look for it by a variety of different methods. Yeah, and this is just one lot. 53 different types of tumors, including benign tumors and pre-invasive cancers, have been shown to be multiclonal. The reason that so far tumors have been shown to be clonal is probably because the way that they are investigated by sampling, nobody's really running around sampling tumors all of the time. Maybe that we are missing the clones. I think I will skip this story because this is only about it, but let me just tell you how, if you really look at it, you will find that a tumor like a malignant mesothelioma, which is an industrial, pollution disease, which, you may have actually field effects, are actually composed of clones, okay? Multiple clones, not one clone. Similarly, low-grade endometrial carcinomas, most of them were found to be clonal, but there were 2 cases, and that is only in a small number that were investigated, that actually seemed to be part of the tumor, same tumor, but had 2 different clones in it. And what is the actual reason to even think about this? Oh, what does it matter? Okay, it may be 1 clone, it may be 2 clones, maybe 5 clones. But if these clones have got different molecular mechanisms and are going to evolve in different ways, they may actually behave not as one cancer, but they may behave as more than one cancer, has been so elegantly shown in this particular paper. In fact, they ask the question, okay? With these multiclonal colorectal cancers with divergent histomorphological features and divergent RAS mutations, are they one cancer or are they separate cancers? And I think we need to really think about it. And I think a lot of what we expect, or we were taught to expect, may not actually be true. And I really do feel that adherence to the idea that a growth must develop from a single progenitor cell is no longer valid, okay? I think it has outlived its usefulness. You may talk about multiclonality, you may talk about clonal heterogeneity. One gives rise to the other when you see 2 different sets of clones, or maybe 3. Have they all arisen from one clone, or were they they were multiclonal right from the beginning? Okay? Now let's come to this last one, about the consistent differences between normal cells and cancer cells, okay? And that you can define a cancer of a cell by and of itself. And here are the instances of cancers that can be normalized. I'm not going to talk about, teratocarcinomas and whatever we call high-grade germ cell tumors. You know that they become teratomas. This has become more and more common these days, and that's one way of getting a tumor to neutralize itself, or to neutralize a tumor, is to make this tumor differentiate. And we're doing it. We're doing it all the time. We're doing it with neuroblastomas. We're doing it with germ cell tumors. But the important thing is, look at the second line over there. You can create a teratocarcinoma. How? By introducing you can normalize tumors. You take teratocarcinoma cells and put it into a blastocyst, a mouse blastocyst. And do you think that blastocyst becomes malignant? No. The teratocarcinoma cells get normalized, and you can find that they actually form parts of different organs in that mouse. You can't do it in humans, but it has been done in mice, and if it can be done in mice, you have to take that experiment and think about it. It's quite possible that this is also happening in higher orders of mammals, okay? You take malignant nuclei, and you take out the ovum, the nucleus of an ovum, and you put that malignant nucleus into that ovum, and what do you get? You just get normal embryos. So what happened? The malignant nuclei just sort of became benign. It was from a malignant cell. So, and we are all the time told that the nucleus of a malignant cell is malignant. That's where all the malignancy is happening. That's where all the abnormal genomes are. So what happens then? What happens to those abnormal genomes when you put it into a perfectly normal ovum, which its own nucleus has been removed? No, just the same way that we do it in our, the way that we are cloning animals and other things. You take an enucleated ovum and you put a nucleus into this. Now, this is even more interesting, that you take normal cells and you put them into abnormal sites, and lo and behold, you get a cancer. It is a completely normal cell. There was no question of its nucleus being malignant and all. And why should we worry? Just look at that last line. Okay, cancers can be produced by inert foreign bodies of a particular size. So there is nothing wrong with the organ, and you just put that foreign material over there. And I'm sure all of you have heard of ALCLs, that's anaplastic large cell lymphomas, that have arisen around silicone breast implants. How did that happen? Well, they're all normal cells over there, and they have, it's not become a breast cancer. So it is not as if it is promoting the breast to become cancerous. There's an ALCL that is occurring around these implants. So normal tissues can be induced to form cancers just by putting inert substances which are not anything to do with this. Dormancy is the same thing. You have the cancer cell, it's sitting somewhere. What do they tell you? Oh, this cancer cell has gone into G0. Yeah, but what made it go into G0? Ever thought about it? It probably was made to go into G0. Yeah, and what made it go? Okay, you never get rid of the last microbe. That means you never get rid of the last bacterium, and yet you get over your infections. You never get rid of the last cancer cell, and yet cancer cells go into dormancy. So there are a lot of people now who are talking about the fact that there is nothing really about not necessarily about the cancer cell that is abnormal. It may be that cancer is a disease of tissue organization, that it is everything that keeps the cancer cell, whatever it is, the cell under control, just as in normal differentiation. And this is called the tissue organization field theory, okay? And this is it, that the normal state of any cell is proliferation. But cells, we don't see cells proliferating all the time, so what is keeping those cells in check? Obviously, crosstalk, cells talking to other cells, and the stroma, which is also cells, talking to one another, okay? That's what is keeping tissue organization intact. So it's not about the cell, it's probably about tissue organization, okay? And just think of this, no? We are all multicellular organisms. I told you, we are metazoa. And in metazoa that have tissues which are organized, what cells are given the right to reproduce? It's only germ cells. Yeah? Otherwise, reproduction in all cells is kept tightly controlled. Only when you need cells to be replaced, aging, repair, then those cells are allowed to, you may call them tissue stem cells or whatever. They are tightly under control by a variety of different things, and only then are they allowed. Now, what happens if the controls, disappear? It's like you haven't you seen, and I'm sure many of you have got people whom you know who are, in your society, and they're very obedient, and they're very good, and they obey their parents, and they do everything that, you expect them to do in society, and then they go abroad, and there, all norms are relaxed. It's a more permissive society. And what does that individual do who was very nice, very obedient, very everything? In that society, he goes he, he starts frequenting bars, he drinks, he does, and people say, how did such a good guy become such a rogue? He didn't become a rogue, okay? It is the permissiveness of the society in which he was that allowed him to express what otherwise you had kept, inhibited him from doing. So what is it that we are talking about? And here, the high priests suddenly realized that the biology of tumors can no longer be understood simply by enumerating the traits of cancer cells, but must encompass the contributions of the tumor microenvironment. So now the tumor microenvironment. And just to paraphrase President Clinton, when they were asking him how he was going to win the election, and he said, it is about the economy, stupid. So this is not necessarily about the cancer cell, it is about the environment, stupid. So which of these things is correct? SMTOT of, and I think, this was a famous joke once upon a time, that here is this young man who has been called to have breakfast at his lordship's place, and he's been served a bad egg. Not deliberately, but I think he got it. It was stinking. So his lordship said, I'm so sorry, I think you've got a bad egg. And you know what he said? No, he didn't want to hurt his lordship. So he said, oh no, my lord, I assure you that parts of it are excellent. So it's parts of SMT and parts of TOFT are actually correct, okay? They are not mutually exclusive. And how do we put this together? And I think we put this together in the evolutionary perspective, okay? And this is about adaptive oncogenesis, and we should understand this. And you have natural selection, acts on germ cells, okay? That's why there are little variations in the germ cells, and then you get small phenotypic variations in the whole, whether it's animals or humans or whatever else, and then it's the environment that decides which of these variations will form the largest, most stable population, okay? It's the same thing that is happening in somatic cells, yeah? They vary a little bit because you've got mutations, etc. And as long as that environment remains the same, nothing much is going to happen. You change the environment, and what appears to you as a normal cell may actually become something that, as I told you, happens to a number of our kids who we think are very good, etc., until they go abroad, okay? Now, so this explains age. Why do we get so many tumors as we age? The reason is simply this, that the environment no longer is stable. When we are young, natural selection works to make reproduction important, okay? We as young people, we have to reproduce. That's the law of nature. We were not, when I'm talking about humans, meant to live as long as we have done. Okay, so that environment in our bodies and what's around us is getting destabilized. Some of us also smoke, some of us do worse things. Okay, like I'm terribly fond of sweets, okay, so I'm indulging, and this indulgence changes the environment in which, why is it that breast cancers are becoming so common now with a westernized diet, etc.? Just think about it, okay? So now, once again, Hanahan and Weinberg, they said what they said, and I will tell you, it is the environment, stupid, okay? Now, why should this matter? Okay, you sit in your armchair and you cogitate and you think, what we have learned about cancer, we have to unlearn. No, it's important because it's about approaches to therapeutics, approaches to prevention, and approaches to cancer research, okay? Today, I'll just read this line because if there are any medical oncologists, you might agree with me, with this person, okay? What does it say? The prevailing clinical paradigm is to apply the most effective agents as the first line of therapy until resistance develops. Then switch to the second line of therapy and repeat the cycle until the patient's demise or the clinician runs out of options. From a selection-centric perspective, that is our evolutionary process, this approach maximizes the probability and minimizes the time necessary for the selection of resistant phenotypes. And therefore, if you are going on this SMT theory, you are not heeding, or you are destroying the environmental controls that are wanting to keep this tumor in check. And now we have immunomodulatory therapy, which is good, low-dose angiogenic therapy, which is dealing with what? Not the tumor, but the environment. Metronomic therapy, okay? Giving low doses continuously so that you're allowing the microenvironment to regenerate and use its influence on the tumor. And of course, targeting intratumoral. So, we also use a whole lot of inflammatory, you know that now people are using, when you have this Barrett's esophagus, you can use anti-inflammatory agents to delay the onset of cancer or to prevent it completely. And you know that we treat a lot of things like H. pylori and hepatitis B and C to prevent the cancers from occurring. And once again, Weinberg, who has now become quite philosophical about the fact that we don't really understand that, you know, cancer is about cells. And he should have been listening to Smithers. You know, in 1962, what did David Smithers say? He said, cancer is no more a disease of cells than a traffic jam is a disease of cars. Okay? A lifetime of study of the internal combustion engine would not help anyone understand our traffic problems. Yeah, because sometimes the traffic is caused by a breakdown in a car, sure, or breakdown in more than one car. But you don't think our nakabandhis and the bad weather and everything, anything to do with the car? No. So if a car is a cancer cell, it's not necessarily the cancer cell that is going to cause accumulation of cars, which is your traffic jam. It could very well be the environment. So I think that Smithers was onto something all those so many years ago. So now I've told you what I have unlearned. Okay? So what have I learned about cancer? And I've learned this, that cancer is a process, and I really do believe, by which an organism-like phenotype evolves over time. It's almost like we are parasited by ourselves. Okay? Cytology and morphology do not define cancer. I honestly think so. It's behavior that does. Cancer may never be defined at the molecular level because there's no fixed molecular thing, and please don't tell me that, oh, we've got this fingerprint and that fingerprint for these cancers. I've already told you that doesn't matter, okay? Yes, it might define a growth, but not necessarily the cell, because I can run around with that fingerprint and still not get the cancer. And cancer research has taught us more about normal biological processes than about cancer itself. Yeah, that's why we haven't gone anywhere on this war against cancer, despite the fact that we've spent billions of money, and certainly the United States has. Okay, and they're talking about moonshots now. I sincerely believe that cancer is about us. It is about our attitudes to life and our lifestyles and the environment. Okay? It may be one of the consequences of senescence. That is a life that is lived too long. And I'll just end with this. Every sort of contradiction can be found in cancer. Cancer is like life. If you think you have understood it, then please think again. Thank you very much indeed.